Okay, so now we're done with immunodeficiencies. We're going to talk about how we are able to diagnose certain immunological diseases through immunodiagnostic technologies. This will probably be split up into two separate parts, so I'd recommend watching both of them if you want a full overview of these technologies. So a quick recap of the blood that we went over a bit when we were talking about serum sickness with regard to type 3 hypersensitivity reactions. So the plasma is basically just the whole of the blood minus the erythrocytes, leukocytes, and the platelets. So that would be the blood with all of its kind of antibodies, its clotting factors, the plasma, proteins. We're just taking away red blood cells, white blood cells, and platelets. And then the serum is just the plasma minus the clotting factors. There are many immunodiagnostic techniques that say different labels. An enzyme label, so basically we're using these different kind of labels to determine the presence of a specific protein or antigen within a blood sample. We're going to go over these in more detail as we progress through these couple of videos. So the main three are the ELISA, the radioimmunoassays, and the immunofluorescence assays. So immunoprecipitation techniques include so they're used to isolate and identify a single antigen within a group of mixed antigens. So you're trying to identify a single antigen within within something like the blood, which has many different kinds of antigens in it. Precipitant antigens could be chromatin, proteins, protein complexes, or tag proteins or RMPs. A precipitation antigen can be separated from a mixture, like the blood, using an antibody that is specific to that antigen. So obviously that antibody is going to target that antigen, and it's going to kind of make it heavier because they're going to form an antigen-antibody complex. And then these complexes are going to settle at a particular layer in the mixture in the test tube, usually at the bottom, because these complexes are going to be heavier than other stuff in the blood. Immunoassay tests use antibodies and antigens to detect the measure or concentrations of macromolecules in a solution. These are the main three types that we're going to talk about: the ELISA, the lateral flow test, and the filariasis test in this video. So the filariasis ELISA, we're going to talk about that now. The specific type of ELISA that we're going to be talking about is a sandwich ELISA. So the monoclonal antibody, OG4C3, is immobilized in the wells of the ELISA plate. So if you look to this image on the right here, you can imagine this black line at the bottom is the ELISA plate, and the monoclonal antibody is the green antibody shown at the bottom. Remember when we talked about immunoglobulins in more detail, monoclonal antibodies are usually produced in mice, and they are all specific to the exact same antigen. So the monoclonal antibodies that we'd be using here would be specific to the antigen that we're trying to assay in the patient's blood, which would be this OG4C3 antigen for the filariasis ELISA. So patient's blood would be added to the well, and antigens in the blood would bind to the monoclonal antibodies. So you can see that by this little red circle bound to the green monoclonal antibody on the diagram. A polyclonal antibody would then bind to this antibody-antigen complex, the polyclonal antibody being this yellow antibody, and then this antibody-antigen-antibody complex would be identified through the use of a secondary antibody tagged with an enzyme. So you can see this purple antibody at the top would be the second antibody, and it would be tagged with this little orange enzyme at the top of the Fc region up here. This enzyme would break down a specific substrate, inducing a color change in the well. And if you see this color change, then it means that you've had this whole antibody-antigen-antibody secondary antibody complex formed, so that these second antibodies tagged with the enzyme can actually break down the substrate. So the substrate would be added to the well, and it would be broken down only if this whole sandwich ELISA complex that we see on the right has been formed. If there's no antigen, if there's no, like, this little red circle, then the polyclonal antibody isn't going to attach to the monoclonal antibody, and the secondary antibody isn't going to attach and produce the substrate change, because every time, after every step, you would have a wash step where you wash the well, and if there's no antigen in the blood, then the polyclonal antibody and the secondary antibody tagged with the enzyme would just be washed away in the wash steps. So color change would indicate that the antigen is in the blood sample. There are 4 different kinds of ELISA test: the direct ELISA, the indirect ELISA, the sandwich ELISA, and the competitive ELISA. The most commonly that I've seen used is the sandwich ELISA, which we've talked about here for the diagnosis of filariasis. But I've also used a sandwich ELISA to diagnose to make a prognosis for systemic lupus erythematosus, because if you just replace the antigen with an antigen for lupus, like a nuclear antigen from a patient's DNA, then it could test for lupus if you wanted to. So moving on to filariasis lateral flow immunoassays. An example of a lateral flow immunoassay is an ICT for filariasis. Filariasis is just a kind of nematode infection. The immunochromatographic test, the ICT, detects small amounts of the antigen produced by these nematodes in the blood. A monoclonal antibody specific for a pathogen antigen is mobilized in a test line. The sample pad is loaded with polyclonal antibodies tagged with colloidal gold, which produces a color. When blood is added to the sample pad, the antigens form complexes with the polyclonal antibodies. These complexes travel to the test line, where the monoclonal antibodies specific to these complexes recognize and bind to them. These ICT tests, commonly referred to as lateral flow, are cheap and easy to perform, far cheaper than PCRs. It's kind of similar to a sandwich ELISA in the sense that we have these monoclonal antibodies at the test line. If you just cut out the yellow polyclonal antibody in this image, and you imagine if it was just 2 antibodies, the monoclonal antibody at the bottom, the antigen, then the polyclonal antibody with the colloidal gold for the sandwich ELISA, it would be the exact same thing, and it's just going to produce a line at the test strip line. And if a line is produced, then that's going to it's going to imply that the antigen is in the blood, and you're going to test positive for the filariasis infection if you've got the nematode